Michael Chapman from Genova Diagnostics, a leading expert in product innovation. He holds a naturopathic doctorate from Bastyr University, contributes regularly to medical texts, and is a familiar voice on the Lab Report podcast. He's here to share his insights and knowledge on how big data intersects with the microbiome and is revolutionizing our approach to health diagnostics. At the end of his presentation, if time allows, Dr. Chapman will be fielding questions from the chat, so feel free to add those in before the end of his presentation. Please join me in welcoming Dr. Michael Chapman.
Great. Thanks so much, Lisa. And just a big thanks to everyone on the FullScript team for giving me this opportunity to speak today. I'm super excited to be here. I think hopefully you'll find this topic really interesting. Going to be talking about the microbiome, but a little bit more around how we need to think about the microbiome moving forward and how we can kind of crack this code, and hopefully how big data, machine learning can really help us along the way. Here's a little bit of a disclaimer. I am the Director of Product Innovation at Genova Diagnostics. And so just to have you all know that right up front. So let's just get into it. You know, it's interesting. I've been finding recently that a lot of my presentations are growing to be less and less about getting into the weeds of a certain biochemical pathway or a certain gut microbes toxin production. I think a big reason why I've been taking a less microscopic view and focusing more on the bigger picture is because as a functional medicine practitioner, I sometimes worry that we can develop a not seeing the forest for the trees type of an issue. I mean, each one of these microsystems is a tree in the forest, or maybe like one of the overpasses on the highway here. But we get so hyper-focused on parts that sometimes we lose focus on the whole. And when we lose that focus as practitioners, we end up with such confused message to the patients that I think sometimes we don't know, and the patients don't know what path we're actually taking toward healing. And this is often demonstrated even in the peer-reviewed literature, where you have a plethora of information, but yet almost any assertion put out in a publication, you're going to find a 180-degree rebuttal somewhere else. Like, literature is supposed to help us differentiate fact from falsehood, and yet we're at a stage where us, as people interested in healthy living, we can't even agree between plant-based diets versus animal-based diets. And so this is no different when it comes to our understanding of the most complicated and dynamic features in our entire human organism, the microbiome. I mean, our understanding of the gut microbiome is a perfect example for the forest versus tree conundrum, because it's so easy to get really compelled with an individual bacteria's role on our health that we tend to focus on bugs like lactobacillus or bifidobacter. But then when we look at the literature, we're surprised to see that actually the results are mixed when it comes to whether they're even associated with good or poor health. So I think today's seminar is all about looking toward the future of our functional and integrative medicine paradigm, and to truly move our knowledge. And by knowledge, I mean what we know that is likely true or not true. We need to use a new set of questions, a new model, and a way to create the research that can predict complex systems rather than simple controlled experiments. And if we just put the microbiome aside for 1 second and look over at how nutrition relates to cellular health, again, it looks immensely complicated. However, the rules for this system are knowable, and it's a complicated program for sure, but it is one that we can actually build into an algorithm, and we can use our understanding of these metabolomic rules to generate our recommendations for things like nutritional supplementation, which is what we do oftentimes on our testing here. It's almost as if we can model this complex system into one large algorithm. And I think this is one way that the future of metabolomic lab testing will continue to be refined. I mean, frighteningly, I'll say this diagram on the screen is actually greatly oversimplified, but the point is that the biochemistry can serve as a model for future machine learned algorithm development. And we will need the power of what today's machine learning can provide, especially if we're attempting to put all the pieces of this immensely complex system together. Questions like, how does a person's genomic code impact their metabolomics and affect their need for certain nutrients? Or how might a person's nuanced diet change the composition of the microbiome in a way that actually generates the symptoms they're experiencing? Like, those questions are difficult enough to answer, but when you start to layer these questions on top of each other, it becomes something like imagining 10th dimensional chess, right? But the power of AI and machine learning is that this daunting task does become more and more possible. However, there's a large caveat, which is this. We have to be very careful about how we train these systems and what questions we ask, because a vague or a biased question could ultimately lead to very interesting conclusions, but alas, conclusions that are ultimately incorrect. So I'd like to show you some examples of what I mean about the importance of asking the correct question, getting a clinically useful answer. Today, my portion is about insight into the microbiome, but it's important to understand that the microbiome is a system within a system. And this goes back to the complexity of putting the puzzle pieces back together. The GI tract's function is threefold. We use kind of this DIG model, digestion, inflammation, or immune response in the gut microbiome to paint the picture of the core functions of the GI tract. And the microbiome is just one piece of this system. And I'll say that it is important to understand a little bit about the methods that are used to assess the microbiome. There's lots of different technologies that can be used. There's a culture analysis, which has its pros and cons. It can detect viable living organisms, which is helpful. There's qPCR, which provides quantitative data for analysis, which is a pro, but it does require an individual developed probe for each organism. And there's even things like whole genome sequencing or shotgun sequencing that it identifies the entire microbiome or nearly the entire microbiome. It can provide insight into what the microbiome is doing by looking at the production and the degradation. But it is also limited in what library you're comparing to, what patient population is used to set those ranges. So I think the point I'm trying to make is the data is only as good as the details and the precision that's used in the testing. And also, the insights you can get from data analysis is only as useful as the question you're trying to pose. And let me explain a little bit. Here's an example of one very basic question that can be asked, and actually is often asked between us clinicians or maybe even the health influencer community. It's not a bad question, but it's limited in its precision. Is there a healthy microbiome? In trying to get more insight into this question, we actually looked at the PCR commensal bacteria profile of over 100,000 patient samples, and we cross-referenced that using survey data, diagnosis criteria to analyze their patterns in the commensal profile. And what we did was, first, to try and answer, is there a healthy microbiome, we created two strongly correlative algorithms that was able to distinguish healthy individuals from patients suffering from the most common GI condition, which is IBS. So one of those algorithms was called the healthy pattern continuum on the left, and the second is called the reference variance score on the right there. And so you can see that those were both statistically significant, but we took it a step further and we plotted these axes in a two-dimensional cross plot. As you can see on the screen here, the green line and the red line are two margins that can be drawn based on the percent of individuals who are either in the healthy cohort as compared to the IBS cohort. Now, that might be interesting enough on its own as a graphical depiction of the risk of IBS based on somebody's microbiome. However, going back to the beginning of my talk, I'm trying to point out that ultimately, understanding what the pattern is telling you is how we're going to make a greater impact on the future. Because this slide is showing that the algorithm that was made to separate IBS patients from healthy patients was completely mirrored when it comes to patients who suffer from chronic fatigue syndrome. Not only that, it was completely mirrored in people suffering from autoimmune conditions. In fact, when we looked across about every major chronic condition, such as mood disorder, heart disease, diabetes, what have you, the pattern here looked identical. And so what that's telling me is this microbiome can be understood in a completely different way. This was just one of the first steps that we took with layering in machine learning into the GIFX profile, but it wouldn't be the last, because we know that the future of understanding the microbiome connection is not in looking at an attendance list. Okay, the days of this bacteria is good, you want more of it, or this bacteria is kind of not good, let's kill it with antimicrobials, that's not the future. And it's a good thing, because those approaches often have even more negative clinical outcomes than positive ones. Because the truth of the matter is that the microbiome assessment itself as a test is a lagging indicator. Why do I say that? The microbiome is responsive to the things on the left. And so if a doctor wipes out someone's microbiome with oregano oil or an antibiotic because they're worried about a potentially pathogenic bacteria, what happens when that person leaves the office? Unfortunately, oftentimes they go right back to the diet, the mood, the sleep patterns that created a hospitable place for that bug they just targeted to kill. Except now that person has even less diversity in their microbiome to counterbalance it. And right, they have less resiliency. So we see this pattern all the time. And not only is the microbiome what I call a responsive system, it's dynamic. What I mean by that is what causes physiologic changes in our body are the things on the right-hand side, which are the products of the microbiome. And there's never one bacteria that's responsible. There are so many different bacteria of different groupings that are all making these things at the same time. So my point here is that the future of microbiome assessment is going to have very little to do with what bugs are there or not. It's going to have everything to do with the patterns they create. And that makes sense because that's systems biology in a nutshell. And if you recall from earlier, the first question you could ask is there a healthy microbiome? Well, we have some data to suggest that there are potential ways to answer that. However, we wanted to start to become more precise about these microbiome patterns. And so our next question was, is there a pattern within the microbiome that makes it more likely to create inflammation? So to answer that question, we actually developed two studies. The first one was an internal study using about 7,000 patient samples. And we tried to correlate those to markers of inflammation in the gut, right? We tried to say, what does the pattern look like when people actually have inflammatory biomarkers? And so what we found was that we could actually predict a certain microbiome pattern that would raise people's or be associated with an elevation in inflammatory biomarkers. But we didn't want to just stop there. We wanted to make sure that it worked in real life. So we partnered with UCLA Medical Center, and we were able to launch a secondary study that actually showed and demonstrated the power of being able to predict people whether they had ulcerative colitis or Crohn's as compared to a functional bowel disorder like IBS. And so it did work. We also went on to look at what's called the methane dysbiosis score. Very similar exercise, but here what we actually did was we looked at GI profiles in their microbiomes at the same time somebody had run a SIBO breath test to try and say, what is the pattern of bacteria that might actually lead to a production of methane? And so we were able to do that exercise as well. And what we find oftentimes when we do these data analyses is that not only are we learning and predicting and developing better assessments, but we actually are finding novel insights. Because one of the things that we know is, yes, methane can cause things like constipation in the GI tract. And there's even a new diagnosis code called intestinal methanogen overgrowth. But another thing that we found was that methane was actually associated with a decrease in levels of the immune markers, such as eosinophil protein X and fecal secretory IgA. So our question became, well, what's going on here? Because not only that, these patients actually had a higher propensity to being infected with potentially pathogenic organisms like blastocystis and Entamoeba histolytica. So that caused us to dig even more into the literature, where we've actually started to create this hypothesis that it makes a lot of mechanistic sense that actually methane has a secondary function in its relationship to serotonin, and that methane production has been associated with decreased serotonin production. This does actually make sense because serotonin creates motility. Maybe this is one of the reasons why people with high methane have constipation. But serotonin's other role in the GI tract is actually to mediate intestinal immunity, including recruiting some of our immune cells to fight infection. So this is a novel insight that some of this big data analysis can actually provide for us. But we didn't want to stop there, actually. We created another two-dimensional cross plot to group people into these 4 zones. You can see at the bottom, we used that inflammation score, and on the left-hand axis, we used that immune suppression score, the methane score. And These zones are basically taking someone's microbiome and categorizing it into the likelihood and its ability to create inflammation, or its likelihood to create methane, or even both. And this can be clinically informative all by itself, of course. But I think one of the other things is like where we don't stop is what else can we do with this? Because I spoke at the top about it being one type of effort to put the complexity of the microbiome back together, but it's an even larger challenge to layer that on top of nutrition. So how about this question? Is there something about a person's cellular footprint that might be suggestive of a pattern in their microbiome, right? If we look at all of their data from the perspective of their organic acids, their amino acids, their fatty acids, is there something there that we might even learn about a prediction on what's going on with their microbiome? And again, we need the power of something like machine learning capacities to help us answer these types of questions. And we also need the expertise of well-seasoned statisticians and data analysts to help make sure that we're actually formulating the questions correctly. Otherwise, it can be kind of treacherous. So one thing is, I'm really excited to say that we're already working on this and many other similar questions. The insights that we are gleaning from this is really incredible and demonstrates the power of what we have going on here at Genova. I'm showing you here, what I'm showing you is a plot of actually the predictive power of just one individual biomarker from our nutritional testing. So what you can see is as this biomarker goes higher or lower, the likelihood that their microbiome is more inflammatory changes, the likelihood that their microbiome produces more methane changes, because what you see is those 4 different colors reflect the propensity or the risk that they're going to show up in one of those 4 zones. And while that's cool that one biomarker can do that, what do you think happens when you put together 50 of the most predictive biomarkers? Yeah, what you have is a very, very strong algorithm that can actually provide a high level of risk prediction on what that microbiome might be doing. And remember, this is not even coming from a microbiome assessment. This would be coming from something like a nutritional assessment, organic acids, amino acids. So that type of analysis is not only clinically useful, it's also cost saving. And it tells us novel insights into biomarkers that we would never have even connected with the microbiome before. So for example, some of the markers might actually be fatty acids that we can't even think even have a connection to the gut production or the microbiome. It gives us new avenues to learn, and it gives us new scientific insights, which is how you can see this entire cycle just keeps moving forward and forward. And so that is kind of a little bit of the sense that I wanted to talk about today, which is moving from this idea that the microbiome is all about these bugs that are sitting there in there doing things. Maybe they're making different positive products like short chain fatty acids. Maybe they're making inflammatory components like LPS. And while that is one way to look at it, we're going to be able to understand a lot more in the future by thinking that this is an entire ecosystem that shifts and grows and shrinks. And those types of larger concepts are where we need really powerful tools to help us put those back together. And once we do that, we can even move this and start layering all those different omics that we're thinking about, the genomics, the metabolomics, all of it starts to fit together. And that's kind of the vision of where I see and where we at Genova see this functional medicine paradigm moving. And so hopefully you found some of this information useful and that it gave you a little bit of a sense of where we're headed when it comes to the future of gut health testing and how it's likely to just continue to evolve in new ways as it always does, and ways that we never saw possible. So I'm going to pause there. I just want to say thank you so much for your time and attention. And I have a few minutes to open it up to the virtual floor for any questions, if there's any questions out there. So thanks again.
Thank you so much, Dr. Chapman. We're now going to start our Q&A portion of the session, and Dr. Chapman will be now answering a few questions from the chat. To get us started, I know that the microbiome is always changing. There are so many things that can impact someone's microbiome that they encounter in their everyday life. Can you guide us on when we should start thinking about retesting someone's microbiome? When should we be thinking that it's time for a new test?
Yeah, we sort of have like a stock thought on that, but then there's the caveat that, as you said, it does change in response to, because like I said, it's a lagging indicator. Things like, does somebody's mood change, does their sleep change, does their diet change? So I tend to think that anytime we're making a dramatic change clinically or therapeutically, then a good window of time is, maybe 3 months. If somebody's more on a little bit of just kind of a routine maintenance, then we might be thinking about every 6 months, or maybe you could even potentially push that out to a year if you're not actively working on something GI related. I think, but obviously that's all very malleable based on the patient in front of you.
All right, thank you. We're getting some questions in the chat about which tests are these types of analyses available on. Maybe you can talk a little bit about what folks in the audience should be looking for when they're looking at Genova's test menu, if they're interested.
Yeah, sure. So a lot of the graphics that I showed earlier, especially around how we are using the microbiome information to create sort of these larger data analysis, pattern analysis, that's going to be available on our GIFX comprehensive profile. And so that's probably where you're going to be wanting, if you're wanting to have those analyses run on your patient. I think one of the other tests that I demonstrated there was what's called the NutriVal profile, or very similar test is called the metabolomics, which is more of an at-home version of the NutriVal. So those are kind of the two places that I would be looking for the information I showed here.
Thank you. All right. I'm surprised this is not the very first question that we got. I have a question about sample collection, if you're willing. Someone has made the astute observation that it's really important. It may be important to take a few days of stool samples to get accurate results. Maybe you can comment on how best to guide patients to collect a sample for accurate results for this type of test.
Yeah, very good. So we have, for that GIFX comprehensive test, we have a 1-day option available and we have a 3-day option available. A lot of patients obviously prefer the 1-day option for obvious reasons. But if a clinician is suspecting any type of parasites, I think that is the main place where we make a differentiation. If there's a suspicion around parasitic infection, we do get a little bit better recovery of those organisms with the 3-day. We've done some internal analyses actually based on how we are processing them. And we have kind of a unique method of how we do that. And so we were able to determine that parasite recovery was about 90% with the 1-day test, and that 3-day gave us a little bit of a 5 to 10% improvement on top of that for parasite recovery. If you're not suspecting parasites, they're not showing signs and symptoms, then oftentimes clinicians will use the 1-day because it's easier on patients.
Awesome, thank you. I apologize if you're hearing some background noise through my feet. Last question, what is the approximate, this might be more questions, sorry. What's the approximate timeline for some of those exciting advancements with disease correlation? Or is this already available for providers?
So what we showed from a GI pattern analysis is absolutely already available on the GIFX comprehensive. The timeline around some of the things that I teased, I would have to leave as a TBD at this point. There's a lot of, as you can imagine, with big data analysis and machine learning, the analysis portion of it is very, very exciting. And then you have to get into the validation part of it, which is probably even more important, right? Because you want to make sure that everything is exactly correct when it's coming out the door. So at this point, I would say that's a little bit of a TBD, but it's definitely something that everyone here is very excited about. And like I said, it's actually one of many questions that we are constantly working on. So I think the future is incredibly exciting. Okay.
I think we're going to close it there. If you've got questions in the chat that haven't been answered, we'll reach out with an answer for you. So now is your chance in the last few seconds to throw your questions in the chat. As mentioned, the session is recorded. You'll get a recording of that and also the slides. We had a few questions about that as well. So please join me in thanking Dr. Chapman, and thank you all for joining us today for this really interesting topic. The next session starts in 5 minutes, and you can join us there by clicking back to the schedule tab. I hope to see you all soon. Thank you so much, Dr. Chapman.
Thank you.